Hello friends, in this video we are going to discuss about translation in prokaryotes. In this we will discuss about definition of translation, then we will discuss central dogma. We will discuss components of translation like messenger RNA, transfer RNA, ribosome, enzymes and proteins. We will discuss about initiation factors. Then we will see the synthesis of aminoacyl transfer RNA. We will discuss about all the three steps of translation: initiation, elongation and termination. Under initiation, we will see the events involved. Then we will see its explanation with the help of diagram. We will also discuss about certain facts and terms. Under elongation, we will discuss about definition of elongation. Then we will see elongation factors. We will see the explanation of elongation with the help of flowchart. We will discuss about elongation process in detail. We will discuss all the three steps involved in elongation. Then we will discuss about EFTU, EFTS exchange cycle. We will also discuss about peptide bond formation. Then under termination step, we will discuss about stop codons and release factors. Then we will discuss about all the events involved in termination: stop codon recognition, release factor binding, polypeptide release and ribosome dissociation. So stay tuned. Now let us see initiation step in prokaryotic translation. Its introduction. Representative prokaryotes, E. coli. So first of all, what is translation or what is protein synthesis? The process by which a cell makes proteins using the genetic information carried in messenger RNA. This messenger RNA is made by copying DNA, and the information it carries tells the cell how to link amino acid together to form proteins. In prokaryotes, it is a tightly regulated process that begins with the assembly of initiation complex. This initiation complex we will discuss later. Now the central dogma. It is a theory stating that genetic information flows only in one direction. DNA molecule makes its copy by the process of replication. DNA then forms RNA by the process of transcription, and RNA then forms protein by the process of translation. So here the information from DNA to RNA to protein flows unidirectionally. There is no complementarity between amino acid and messenger RNA. Hence, translation is not controlled by complementarity but by genetic code. Genetic code we have already discussed in detail in separate video, the link of which is given in the description box. Since amino acid cannot read this genetic code, they are dependent on an adapter molecule called transfer RNA. We have already discussed transfer RNA in separate video. You can refer my video, the link is given in the description box. Now let us see the components of translation. First is messenger RNA. It acts as a template. It carries the genetic information copied from DNA in the form of a series of 3 base code words, each of which specifies a particular amino acid. Therefore, codons specify amino acids. Second is transfer RNA, which is an adapter molecule. It has anticodon for codons of messenger RNA, and these codons are complementary to anticodons. Hence, base pairing occurs between these two, that is anticodon and its complementary codons. Hence, it brings accurate or specific amino acid in accordance with the codon. It deciphers code in messenger RNA, or in other words, it discovers code in messenger RNA. Each type of amino acid has its own type of transfer RNA, which binds it and carries it to the growing end of a polypeptide chain or protein. Then enzymes. Among enzymes, first is peptidyl transferase. It facilitates the formation of peptide bonds between amino acids. It is a component of large ribosomal subunit. Second is aminoacyl transfer RNA synthetase. It attaches appropriate amino acid to its transfer RNA, that is loads or charges transfer RNA. Then third is methionine transfer RNA formyl transferase. It catalyzes the addition of formyl group to methionine. So formyl methionine is the first amino acid of protein or polypeptide chain in prokaryotes. Then ribosome. It is manufacturing unit. It has two subunits, small subunit and larger subunit. Small subunit reads messenger RNA and larger subunit joins amino acid to form polypeptide. Now let us discuss about some of the important aspects of ribosome. Ribosome normally exists as separate subunits, that is smaller subunit and larger subunit. They come together when bind to a messenger RNA near its 5' end. Then it reads the nucleotide sequence of messenger RNA from 5' to 3' direction. It synthesizes protein from amino acid in an N-terminal to C-terminal, that is from amino terminal to carboxy terminal. Now the location of ribosome, it is located in the cytosol, either free floating or associated with endoplasmic reticulum. Now the ribosomal sites. First is aminoacyl transfer RNA binding site or A site. It is the entry site for new transfer RNA charged with amino acid or aminoacyl transfer RNA. The peptidyl transfer RNA binding site or P site. It is occupied by peptidyl transfer RNA or binding site for transfer RNA that carries the growing polypeptide chain. Third is the exit site or E site. It is the exit site for the deacylated transfer RNA, or in other words, it is exit site for the transfer RNA which has done delivering the amino acid. Hence, it is a binding site for transfer RNA following its role in translation and prior to its release from the ribosome. Now, let us see the structure of the functional ribosome. This is smaller subunit, this is larger subunit. This is the messenger RNA. Three sites of ribosome are aminoacyl transfer RNA binding site or A site, this is peptidyl transfer RNA binding site or P site, and this is exit site or E site. Fifth component of translation are proteins. These are soluble factors that are required for proper initiation, elongation and termination of translation. Here we will discuss about factors involved in initiation. Factors of elongation and termination we will discuss in later videos. Initiation factors. Initiation factors in prokaryotes involves coordinated action of IF1, IF2 and IF3, known as initiation factor 1, 2 and 3. Their main function is to assemble the ribosomal subunit around the messenger RNA and recruit initiator transfer RNA. IF1 prevent the premature entry of transfer RNA into A site of ribosome. IF3 prevent premature association of ribosome subunits. Hence, small ribosomal subunit is separated from large subunit with the help of IF1 and IF3. Or in other words, its role is to stop the 30th subunit binding to 50th subunit in the absence of messenger RNA and initiator transfer RNA. Otherwise, their association will result in non-functional ribosome. IF2. It has ribosome-dependent GTPase activity, which regulates and directs the initiator transfer RNA to its correct position in the initiation complex. Initiator transfer RNA is the transfer RNA charged with N-formyl methionine amino acid. When specific amino acid bind to its specific transfer RNA, it is known as aminoacyl transfer RNA. Let us see the synthesis of aminoacyl transfer RNA. It is two-step reaction. First, reaction of amino acid and ATP to form an aminoacyl adenylate or aminoacyl AMP. Second, aminoacyl group of aminoacyl AMP is transferred to 3' end of transfer RNA to form aminoacyl transfer RNA. So the overall reaction is amino acid plus ATP plus transfer RNA in the presence of enzyme aminoacyl transfer RNA synthetase gives aminoacyl transfer RNA plus AMP plus inorganic phosphate. Now let us see the importance of aminoacyl transfer RNA synthesis. To take part in protein synthesis, each amino acid must be covalently linked to a transfer RNA molecule. This depends upon the adaptive function of transfer RNA to ensure that correct amino acids are incorporated. So, in order to take part in protein synthesis, amino acid is covalently linked to transfer RNA. So, the transfer RNA function as adapter molecule and correct amino acid is incorporated. Covalent bond between amino acid and transfer RNA is a high energy bond, and this enables the amino acid to react with the end of the growing polypeptide chain to form new peptide bond. Hence, synthesis of aminoacyl transfer RNA is also referred to as amino acid activation. There are three steps of protein synthesis: initiation, elongation and termination. Let us see the events or steps involved in initiation in detail with the help of this flowchart. Initiation factor 1 and 3 binds to 30S ribosomal subunit. Then it binds to messenger RNA via Shine-Dalgarno sequence and move 3' along the messenger RNA. That is, it moves 5' to 3' direction along the messenger RNA until it locates the initiation codon AUG. Then initiated transfer RNA binds to the initiation codon. This initiated transfer RNA is charged with N-formyl methionine and it is complex with IF2 and GTP. This IF2 facilitates the binding of initiator transfer RNA to the initiation codon. Then initiation factor 1 and 3 are released. Now 50S ribosomal subunit binds with the release of IF2 and hydrolysis of GTP. And this binding of 50S ribosomal subunit to the 30S ribosomal subunit form 70S initiation complex. Then it is ready for next step of protein synthesis, that is elongation. Now let us understand this initiation step with the help of this diagram. Initiation factors bind to 30S ribosomal subunit. First, binding of initiation factor 1 and 3 takes place. Then this 30S ribosome binds to the messenger RNA via Shine-Dalgarno sequence and it moves along messenger RNA in 5' to 3' direction till it locates the initiation codon, that is AUG. So this is the Shine-Dalgarno sequence and this is AUG. Then binding of initiated transfer RNA takes place, which is charged with N-formyl methionine and is complex with initiation factor 2 and GTP. This initiation factor 2 facilitates the binding of initiator transfer RNA to the initiation codon, that is AUG. Then initiation factors 1 and 3 are released. This is known as 30S initiation complex. After that, initiation factor 2 is released and GTP is hydrolyzed. With this, 50S ribosomal subunit comes and binds with this 30S initiation complex. Now it is known as 70S initiation complex. There are three sites: A site, P site and E site. This P site is occupied by initiator transfer RNA. Now it is ready for the next step of translation, that is elongation. We will discuss this step in detail in the next video. Now let us discuss the facts and terms related to initiation. First codon translated in all messenger RNAs are the start codon, which is also known as initiation codon. that is AUG, which codes for methionine. Transfer RNA F-met is used for initiation, and this transfer RNA F-met is called initiator transfer RNA. Transfer RNA M-met is used for internal AUG codons. Hence, there are two different transfer RNAs for two types of AUG codons. In prokaryotes, first amino acid is N-formyl methionine. Hence, aminoacyl transfer RNA used in initiation is F-met transfer RNA F-met. Now, Shine-Delgado sequence. Short sequence rich in purines. 5-AGG, AGGU 3-. It lies 5-to AUG, initiation codon. It is the leader sequence upstream of the first AUG codon, also known as the ribosomal binding site. It is complementary to part of 16S ribosomal RNA in the small ribosomal subunit. Hence, it is base paired to this part of 16S ribosomal RNA. As a result, small ribosomal subunit attaches to messenger RNA at the correct location. So, this is Shine-Delgado sequence, which lies 5-to start codon or initiation codon known as AUG. It is short sequence rich in purine. Its sequence is AGG, AGGU. It is complementary to part of 16S ribosomal RNA. Hence, these two are base paired, as a result of which small ribosomal subunit attaches to messenger RNA at the correct location. So, this is anti-Shine-Delgado sequence present on the 3' side of 16S ribosomal RNA. This sequence is identified by ribosome GTP complex. Why GTP is essential? IF2 undergoes significant conformational changes upon binding GTP or GDP, which is essential for its function as a molecular switch during translation initiation. GTP bound form of IF2 is active and promotes the assembly of initiation complex and thus results in accurate initiation of protein synthesis. GDP bound form is inactive. Upon GTP hydrolysis, IF2 is released and the ribosome is ready for the elongation. Hence, activity of IF2 is strictly dependent on GTP. Initiation factors IF1, IF2 and IF3. We have already discussed in introduction video of prokaryotic translation. You can refer this video. The link is given in the description box. Why does initiator transfer RNA binds to P site rather than A site of ribosome? To ensure that translation begins correctly. This positioning allows the A site to remain empty for incoming charge transfer RNAs, which facilitates the elongation of the polypeptide chain. Now let us discuss elongation step in prokaryotic translation. What is elongation? It is the extension of polypeptide chain by adding amino acid one by one to the growing polypeptide chain. It is the second and crucial step in translation. This process is facilitated by several elongation factors, EF and ribosomes, which moves along the messenger RNA molecule codon by codon. So let us see these elongation factors, or in short EF. Elongation is aided by three protein factors. They are EFTU, EFTS and EFG. Now let us see them one by one. EFTU, that is elongation factor thermo-unstable. It delivers aminoacyl transfer RNA, that is transfer RNAs carrying amino acid to the ribosome's A site, in order to ensure correct codon-anticodon pairing. It requires GTP for binding to transfer RNA and hydrolysis of GTP. Upon correct pairing, that is codon-anticodon pairing, release of EFTU GDP takes place. Now second is EFTS, elongation factor thermostable. it helps recycle EFTU by exchanging GDP for GTP on EFTU. That is, regenerates EFTU after the latter has yielded the energy contained in attached GTP molecule. Now third is EFG, that is translocase. It mediates or facilitates translocation of the ribosome along the messenger RNA, moving the transfer RNA from A site to P site and from P site to E site when the translation initiation complex is formed. This we have already discussed in initiation video. You can refer my video, the link is given in the description box. The transfer RNA binding region of ribosome consists of three compartments. First is A site, that is aminoacyl site. It binds incoming charged aminoacyl transfer RNAs. Second is P site, that is peptidal site. It binds charged transfer RNAs carrying amino acid that have formed peptide bonds with the growing polypeptide chain but have not yet dissociated from the corresponding transfer RNA. Third is the E site or exit site. It releases dissociated deacylated transfer RNAs so that they can be recharged with free amino acid. This is the ribosome and these are the A, P and E sites of ribosome. Now there is one exception regarding the translation. Initiation codon AUG is positioned in P site, hence all except initiating methionine transfer RNA, FMET transfer RNA, FMET occupies P site at the beginning of the elongation phase of the translation directly. Hence vacant A site is ready to accept the transfer RNA for the next codon after AUG, and in this way elongation takes place. Now let us understand this elongation in brief with the help of this flowchart. Elongation begins with the binding of second aminoacyl transfer RNA at the ribosomal aminoacyl site or A site. This is escorted by elongation factor carrying GTP, that is EFTU. As the transfer RNA binds, GTP is hydrolyzed and EFTU is released. Then a peptide bond is formed between N-formyl methionine transfer RNA at the P site and second aminoacyl transfer RNA at the A site, and this peptide bond formation is catalyzed by the peptidyl transferase. Then N-formyl methionine is transferred to aminoacyl transfer RNA at A site, forming a peptidyl transfer RNA at that site, and an uncharged transfer RNA is left at the P site. After that, peptidyl transfer RNA is translocated to P site and uncharged transfer RNA is translocated to E site. A site becomes empty, that is unoccupied. Then new aminoacyl transfer RNA bind to A site and process continues in a cyclic manner till entire messenger RNA is read or decoded. Now let us understand this elongation step or phase with the help of this diagram. These are the A site, P site and E site of the ribosome. As we have discussed earlier that initiation codon AUG is positioned at the P site of the ribosome. Hence, the initiating methionine transfer RNA comes and binds to this P site, as a result of which A site is empty, which is then ready to bind with the transfer RNA carrying the second amino acid, and in this way elongation begins. So the aminoacyl transfer RNA for the second codon comes and binds to this A site, and this is escorted by EFTU GTP, elongation factor thermo unstable. As soon as this aminoacyl transfer RNA binds to the A site, GTP is hydrolyzed and EFTU is released, which has GTP. Then EFTU GTP is regenerated by the EFTU EFTS exchange cycle, which we will discuss in detail in the next video. So what we see that initiating transfer RNA carrying the N-formyl methionine is present at the P site, and the transfer RNA carrying the second amino acid is present at the A site. Then the peptide bond is formed between these two amino acids. This formation of peptide bond is catalyzed by the peptidyl transferase enzyme. After that, N-formyl methionine is transferred at the A site. Hence, peptidyl transfer RNA is formed at the A site and the uncharged transfer RNA is left at the P site. After that, peptidyl transfer RNA is translocated from A site to P site and uncharged transfer RNA is translocated to the E site. This then exits from this E site. Now the A site becomes empty or unoccupied, which then binds with the new aminoacyl transfer RNA depending upon the codon that is present in the A site. So the process continues in the cyclic manner till the entire messenger RNA is read or decoded. Now let us see this elongation process in detail. This elongation process includes 3 steps. First is aminoacyl transfer RNA binding or decoding. Second is peptide bond formation or transpeptidation. And third is translocation. Now let us see these steps one by one. So first is aminoacyl transfer RNA binding or decoding. An aminoacyl transfer RNA selected based on second messenger RNA codon in the A site enters and binds via codon-anticodon interaction with the help of EFTU-GTP complex. This EFTU mediates the entry of aminoacyl transfer RNAs into A site. It binds as an aminoacyl transfer RNA EFTU-GTP complex. First, EFTU binds GTP. This activated EFTU-GTP complex binds to the transfer RNA, that is aminoacyl transfer RNA. So before the process, EFTU-GTP are free. Then EFTU-GTP binds and forms binary complex. Then it binds with aminoacyl transfer RNA and thus ternary complex is formed. This EFTU-GTP then mediates this aminoacyl transfer RNA to the A site of the ribosome. So it binds with the ribosome as EFTU-GTP aminoacyl transfer RNA complex. Binding of the aminoacyl transfer RNA to the A site is driven with the help of hydrolysis of GTP of the complex to GDP. At this point, EFTU is released, leaving the new transfer RNA in the A site. EFTS mediates the release of EFTU-GDP from the ribosome and regeneration of EFTU-GTP. So how the regeneration of EFTU-GTP takes place? By EFTU-EFTS exchange cycle. First, EFTS binds to EFTU and displaces the GDP. Then GTP binds to the EFTU and displaces EFTS. EFTU-GTP is now ready to take part in another round of elongation. Now let us understand this EFTU-EFTS exchange cycle with the help of this cycle. In the process of elongation, aminoacyl transfer RNA is mediated or escorted to the A site of ribosome with the help of EFTU-GTP in the form of EFTU-GTP aminoacyl transfer RNA complex. So once it enters the ribosome, hydrolysis of GTP takes place into GDP and inorganic phosphate, and EFTU-GDP is released, leaving aminoacyl transfer RNA to the A site. Then this GDP is displaced by the EFTS and forms EFTU-EFTS. Then this EFTS is displaced by GTP and EFTU-GTP is regenerated, and it is now ready to take part in another round of elongation. Again it binds with another aminoacyl transfer RNA depending upon the codon present in the A site. and the process continues. Now the second step of elongation, that is peptide bond formation. It is the second step in elongation. It is catalyzed by peptidyl transferase enzyme, an RNA based ribosome that is integrated into 50S ribosomal subunit. In this reaction, the carboxyl end of the amino acid, or in further steps polypeptide chain bound to the transfer RNA in the P site, is uncoupled from the transfer RNA and becomes joined by a peptide bond to the amino group of amino acid linked to the transfer RNA in the A site. And energy for each peptide bond formation is derived from the high energy bond linking each amino acid to its transfer RNA. Now let us see peptide bond formation reaction. The amino group of the aminoacyl transfer RNA attacks the carbonyl group of the ester linkage of peptidyl transfer RNA to form tetrahedral intermediate. This intermediate collapses to form the peptide bond and release the deacylated transfer RNA. Peptide bond formation involves an acyl group O to N migration and converts an ester to an amide. So now let us understand the peptide bond formation reaction. Amino group of the aminoacyl transfer RNA, which is present at the A site, attacks the carbonyl group of the ester linkage of the peptidyl transfer RNA, which is present at the P site, and tetrahedral intermediate is formed. Then this intermediate collapses to form peptide bond. This is the peptide bond, and it releases the deacylated transfer RNA, that is deacylated transfer RNA is left at the P site. Peptide bond formation involves an acyl group O to N migration, and it converts ester bond into amide bond. And this reaction is catalyzed by peptidyl transferase enzyme. Now the third step, that is translocation. In the third step, a complex of elongation factor EFG, which is also known as translocase, and GTP, that is EFG GTP, binds to the ribosome. And after binding, three concerted movements occur, collectively called translocation. First, deacylated transfer RNA move from the P site to E site. Then second, the dipeptidyl transfer RNA in the A site move to the P site. And third, the ribosome moves along the messenger RNA, that is in the 5' to 3' direction by 3 nucleotide to place next codon in the A site. Now let us see some of the facts related to elongation. Elongation proceeds with single movement of the ribosome, each called translocation event. Movement of transfer RNA from A to P to E site is induced by conformational changes that advance the ribosome by 3 bases in 3' direction. After translocation events, the A site is empty and ready to receive the next aminoacyl transfer RNA. A site and E site cannot be occupied simultaneously. Thus, deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation. The energy for each step along the ribosome is donated by elongation factor that hydrolyzes GTP. GTP energy is required both for the binding of a new aminoacyl transfer RNA to the A site and for its translocation to the P site after formation of peptide bond. Elongation continues, adding one amino acid to the C terminal end of the growing polypeptide for each codon that is read, with peptidyl transfer RNA moving back and forth from the P site to A site as it grows. Now one may think, what will happen if there was no messenger RNA? If messenger RNA was not present in the elongation complex, the ribosome would bind to transfer RNAs non-specifically and randomly, and hence specific polypeptide would not be synthesized. Now let us understand this elongation process in detail. Aminoacyl transfer RNA that is selected on the basis of the second messenger RNA codon in the A site enters and binds via the codon-anticodon interaction with the help of EFTU-GTP complex. So, this entry of aminoacyl transfer RNA is mediated by EFTU. Now, a peptide bond is formed between this N-formyl methionine and the second amino acid. And this peptide bond formation is catalyzed by the enzyme peptidyl transferase. Then this N-formyl methionine is uncoupled from the transfer RNA at the P site and it joins to the amino group of the amino acid which is linked to the transfer RNA at the A site by peptide bond. Hence, deacylated transfer RNA is left at the P site and A site consists of peptidyl transfer RNA. Then the translocation process takes place and this is mediated by the EFG and GTP, which is also known as translocase. So, in this process, deacylated transfer RNA now move from P site to E site. Dipeptidyl transfer RNA, which was present at the A site, moves to P site. The ribosome moves along the messenger RNA in the 5' to 3' direction by 3 nucleotides and places next codon in the A site. And technically, we can say that messenger RNA is shifted in this direction and exposes new codon to the A site. Now the A site is empty and it is ready to receive the next aminoacyl transfer RNA. This deacylated transfer RNA exits from the E site. A site is empty and it is ready to receive the next aminoacyl transfer RNA. A site and E site cannot be occupied simultaneously. Thus, deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation. So, A site and E site cannot be occupied simultaneously. Hence, the deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation, and this process continues in the cyclic manner till the entire messenger RNA is read or decoded. So, elongation continues, adding one amino acid to the C terminal end of the growing polypeptide for each codon that is read, with the peptidyl transfer RNA moving back and forth from P site to A site as it grows. Now let us discuss about termination step of prokaryotic translation. It is third and final step of translation. Peptide bond formation and elongation of polypeptide continues until stop codon appear on A site. What are these stop codons? These are termination codons. They are UAG, that is amber codon, UAA, ochre codon, and UGA, opal codon. If stop codon appear on A site, it is not recognized by transfer RNA carrying amino acid because stop codon do not have anticodon. Then how are these recognized? They are recognized by release factors when they enter the A site. Bacteria have 3 of these. RF1, it recognizes termination codons UAA and UAG. RF2 recognizes UAA and UGA. RF3, it stimulates dissociation of RF1 and RF2 from ribosome after termination. Thus, either RF1 plus RF3 or RF2 plus RF3 bind depending on the exact termination codon in the A site. RF1 or RF2 bind at or near the A site, whereas RF3 GTP binds elsewhere on the ribosome. Now the functions of release factors. It causes the peptidyl transferase activity to transfer the polypeptide to water molecule instead of aminoacyl transfer RNA. Thus cleaves the bond between the polypeptide and transfer RNA in the P site. Free polypeptide now leaves the ribosome, followed by messenger RNA and free transfer RNA. Ribosome dissociates into 30S and 50S subunits, ready to start translation again. How ribosome dissociation takes place? Ribosome recycling factor RRF function in ribosome dissociation. This acts together with EFG in a reaction that uses hydrolysis. Ribosome release factor enters the P or A site and unlocks the ribosome. Dissociation of ribosome requires energy which is released from GTP by EFG. IF3 is also required, which prevents the ribosomal subunits from attaching together again. Now let us see the events of termination. First is stop codon recognition. Stop codons UAA, UAG or UGA are recognized by release factors when they enter A site. Second is release factor binding. RF1 recognizes UAA and UAG. RF2 recognizes UAA and UGA. These bind to the A site of the ribosome. Polypeptide release. Release factors mimics the structure of transfer RNA. These have peptidase activity that hydrolyzes the bond between the transfer RNA in the P site and the newly synthesized polypeptide chain, releasing the polypeptides. Fourth is ribosome dissociation. Third release factor RF3 binds to RF1 or RF2 and with the help of EFG and ribosome recycling factor cause the ribosome to dissociate into 30S and 50S subunits, releasing the messenger RNA and transfer RNA. IF3 is also involved in dissociation. Ribosome recycling factor and EFG promote the dissociation of transfer RNA and messenger RNA from the ribosome. IF3 facilitates the separation of ribosome subunits. Now let us understand this with the help of diagram. When stop codon enters the A site, it is recognized by the release factor. Release factor comes and binds with the A site. This release factor has peptidase activity and it hydrolyzes the bond between transfer RNA and newly synthesized polypeptide chain in the P site. Then this completed polypeptide chain is released. After that, components of translation are recycled. Ribosome dissociates into larger and smaller subunits, and this allows the another round of translation to begin or another round of translation starts. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.